Hello everyone and welcome back to our channel Quaint. What if I told you that a 50-cent sensor could detect deadly diseases like cancer and HIV right from your home? No lab, no refrigeration and no doctor needed. Sounds futuristic, right? But it's happening now. Yes, it's happening now thanks to a breakthrough from MIT. And in this video, let's explore this game-changing innovation that could revolutionize global healthcare. Globally, millions die each year due to what? Due to late or no diagnosis. Yes, globally, millions die each year due to late or no diagnosis, especially in remote areas where access to labs and refrigeration is limited. And if you think about traditional diagnostic tools, then traditional diagnostic tools require complex logistics, expensive equipment, and skilled professionals. So in remote areas, these are not available. Again, if you think about war zones, tropical areas, rural villages, okay, means where problems are there, labs are not available, skilled professionals are not available, refrigeration systems are not available. So for those areas, this innovation is so important. Innovation was made by researchers at MIT. So researchers at MIT have developed a DNA sensor, okay, that costs only 50 cents. Only 50 cents, okay. So what is that one? That is actually a DNA sensor. And this DNA sensor uses, and one more plus point is there, no refrigeration is required. Yes, no refrigeration is needed for this innovation. That's why this is so important. And cost is also so low. Means actually you can compare this with a pregnancy kit, home pregnancy kit, you know that thing. That is very easy to use and that is also affordable for almost everyone. So this innovation is almost like that, pregnancy kit. Okay. So no refrigeration is needed. What is that one? That one is actually a DNA sensor. They used an enzyme in this DNA sensor. What is there? An enzyme is there. Name is CRISPR Cas12. It is not Cas9. Okay. In my earlier videos, you must have watched that Cas9 was there. Not must have. I should rather say you might have watched Cas9, okay, and here we are talking about Cas12. So this is an enzyme. Name of that enzyme is Cas12. Better to say here CRISPR Cas12 because Cas12 is related to this CRISPR. Means actually this is a CRISPR associated enzyme, Cas12. So that DNA sensor uses this CRISPR Cas12 enzyme, which is equivalent to biological search and destroy tool. And when it finds the target gene, say linked to cancer or HIV, when this sensor finds the target gene, okay, say for example, the gene is linked to cancer or HIV. That means if the patient has cancer, so gene is linked to cancer. And if the person has HIV, in that case, the gene is linked to HIV. So, and one more thing, the sensor is made for only one disease. The sensor which detects HIV, the same sensor does not detect cancer. Or the sensor which detects cancer, that the same sensor cannot detect HIV. Okay, so say the sensor is made for detecting HIV. So if you have HIV, in that case, that sensor detects that gene. Okay, and in this case what happens? This shreds nearby DNA, and this activity does something special. What is that special thing? This activity means the sensor shreds nearby DNA, so this activity changes the electrical signal of the sensor. And since electrical signal there is a change in electrical signal, that means that confirms the disease's presence. But there was one major hurdle with this innovation. What was the major hurdle? This sensor degraded quickly under heat. But the problem was solved by MIT researchers. They used a protective coating made from polyvinyl alcohol. Okay, and that coating keeps the sensor stable even at 150 degrees Fahrenheit for 2 months. Okay, for 2 months. Not a small time, not a short time actually. Okay, anyway, so the problem of heat was removed by the MIT researchers. So that means what? That means refrigeration is not required. Refrigeration is not required, doctor is not required, lab is not required. And you know that globally many places are there, many situations are there where detection is very urgent, but facilities are not available. So now the scientists are trying to scaling up this manufacturing and trying to making it available globally. And you already know that cost is too low, only 50 cents, half of a dollar. Anyway, so this was all about today's topic, and this is the end of this video. If you found this video interesting, so please like, share and subscribe to our channel. This is the end. Thanks for watching.